Hey everybody, welcome back to another lecture video. In chapter 21, we're going to look at lipids. Now, we have transitioned already from the organic chemistry portion of this class to the biochem portion. And in the last chapter, we were looking at carbohydrates. So we're going to be looking at lipids now, a different class of biomolecule, and I'll be hoping that you guys see or can point out the functional groups that are present in these lipids, and hopefully that ties back to some of the information you guys remember about these functional groups in earlier chapters. Now, let's start just by describing, come on. Is this thing going to work? Here we go. Let's just talk about what a lipid is. Now, a lipid is a heterogeneous class of naturally occurring organic compounds that get classified together on the basis of their common solubility properties. And these guys basically are water insoluble. They are soluble in organic solvents, so we should say organic solvent soluble. And that would include things like diethyl ether, dichloromethane. What am I trying to write here? C2H6O, diethyl ether, CCl2H2, I think it's CH2Cl2, and acetone. Which one's acetone? C3 would be H6O, I want to say. Anyways, these guys would be common organic solvents capable of dissolving lipids. Now, lipids include a few different kinds of molecules. There's fatty acids. There are triglycerides, sphingolipids, phosphoacylglycerols. I don't want to write that one out. Glycolipids. I guess I could say phospholipids. And then we have fat-soluble or water-soluble vitamins. We'll look at a few. We have other molecules called prostaglandins, leukotrienes, thromboxans. We'll look at those. We have cholesterol. I don't want to forget that one. And cholesterol is a precursor to some of the steroid hormones, I guess I could write that, and bile acids. How do you spell steroid? Bile acids are salts that help with digestion. And so we're going to start to look at these different categories, cholesterol included, and give some characteristics of each. Now, triglycerides. Triglycerides are actually a triester of glycerol with 3 fatty acids. And I'm not sure if you can see glycerol there in this molecule. Let's see. Glycerol is this guy. Excuse me. And in glycerol, it's a triol, and so there would be H's on each one of these O's. Okay, that's glycerol. Now, glycerol with 3 fatty acids. Now, fatty acids are long-chain hydrocarbons that have, it's a long-chain hydrocarbon, and then they have a carboxyl functional group. They've got the carboxylic acid. So that's why they're called fatty acids. Now, you guys should remember glycerol being a triol and having 3 OHs on it, alcohol functional groups, and carboxylic acid functional groups react together to form esters. So this is a triester of glycerol. Now, in triglycerides, 2 or 3 different fatty acid components are present. So all the different fatty acids don't necessarily, or I guess what I'm saying, the 3 different the three fatty acids don't necessarily have to be different. They could all be stearates, they could all be palmitates, they could all be oleates. They could also be mixed and have different fatty acids in them. There's a process that we can use that goes, and we actually call it transesterification, and we can go and flip different fatty acids onto the chain if we want to, or onto the structure if we want to. But the difference here that you'll see in some of these groups are that some are saturated and some aren't. So double bond being present in oleate is signified by the fact that it has 18 carbons and then a 1 after it to say that there's a single double bond. Palmitate and stearate also have 16 and 18 carbons respectively, but no double bond, and so the 0 after that is telling you this. Now, the hydrophobic character of this molecule is those long hydrocarbon chains. That's why these things are lipids. That's what makes them water insoluble. The ester groups, although polar, they're buried within the non-polar environment of this molecule, sort of at the center. And since all the parts that are showing are going to be water insoluble, the whole thing just doesn't dissolve. Now, fatty acids, well, the fatty acid component of triglycerides have certain things in common. So I'm going to give you a list here. Practically all of the fatty acids tend to be unbranched carboxylic acids. The fatty acid components of triglycerides all range in size from 10 to 20 carbons. I guess it would help if I... Now, triglycerides sometimes go by other names. I'm trying to think of the other term that people use when they're talking about these. It'll come to me, and then I'll say it. They have even numbers of carbons. So we don't see 19 carbon long, groups. Apart from the COOH that's on the end, they tend to have no other functional groups. Except some have double bonds. We already, well, we should say that. Except because double bonds can count as a functional group. And in most fatty acids that do have a double bond, so I guess I should say, if unsaturated, it's going to be the cis isomer rather than the trans isomer. Now, if you remember a little bit about what the difference is when you have a double bond, right? If the double bond is trans, meaning that the groups on the double bond are on opposite sides, this doesn't really add any kinks to the chain of our hydrocarbon. They can still relatively pack together nicely because they're all shaped the same way. But if you have a double bond and your groups are cis, meaning they're on the same side of that double bond, it leaves a kink in the chain. And the fact that most unsaturated double bonds or most unsaturated fatty acids are cis means that they have these kinks, and that's going to be important. In fact, that's an important physical property of triglycerides, is that depending on the fatty acid present, the fatty acid component, the number of kinks that there are, the melting point of that fatty acid can be affected. So let's talk about the physical properties of triglycerides. melting points of fatty acids will increase. So let's just say MPs go up with increasing number of carbons, more carbons. So a 10 carbon fatty acid or a triglyceride made out of 10 carbon fatty acids will have a lower melting point than a triglyceride made out of 12 carbon fatty acids. So if the fatty acid chains are longer, the melting point will go up. It also goes up as the number of double bonds decreases. I don't like saying it that way. It'll go down, melting points, come on. So more and more carbons makes it go up, and I guess we could say less double bonds. And more double bonds will make melting points go down, and the other thing I guess will be less carbons, if you wanted to be able to study these both ways. And so this has some implications. Triglycerides that are rich in unsaturated fatty acids are generally liquid at room temperature, and we call them oils. Oils have more unsaturated fatty acids. And what we would typically call a fat is going to have more saturated fatty acids. More fatty acids. So again, if they have double bonds, they kink, and that lets them not pack together as well. And so this lowers the melting point and makes them liquid at room temperature in a lot of cases, and so we call those oils. There's a process called hardening, and it's the reduction of some or all of those double bonds of an unsaturated triglyceride using hydrogen gas. And I didn't show you the reaction here, but if we have let's call it a unsaturated fatty acid, and we add hydrogen gas, usually in the presence of some kind of nickel or platinum or palladium, some kind of transition metal catalyst, and this will convert that unsaturated fatty acid into a saturated fatty acid, or it'll convert the liquid / oil into a solid / semi-solid. And we do this for variety of products. For the fats that like Crisco and Spry and Dexo and these others are oils that have been turned into to fats. Margarine and other butter substitutes are often produced by partially hydrogenating polyunsaturated oils that you get from like corn or cottonseed, peanut, soybean, and so on. This process of hardening, though, is the source of trans fats. Now, trans fats are double bonds, again, that are in the trans position, but when this happens because of the process of hardening, or the hydrogenation process, this can lead to fats that do still pack together, but again, these tend to be culprits in our bodies for plaque depositing and that kind of stuff. I don't know the real interactions that the trans fats have in our bodies. If you guys know that, I'd be interested to hear it. I just know that we've associated trans fats with poor health, in the sense that if you have a lot of trans fats, you're more likely to have some kind of issues. But the trans fats themselves come from a process that we're doing. Saponification is the base-promoted hydrolysis of fats and oils. and they typically do this in an aqueous sodium hydroxide or potassium hydroxide solution, so in some kind of strong base. And saponification just comes from the word which basically meant soap in the old days. This is the process that we use to make soaps. We take fats either obtained through the oils or through oils from plants or fats from animals, and if we boil them with this strong base, we produce glycerol, which we can we're basically hydrolyzing the ester bond here. We get back our alcohol or our triol, glycerol, and then we get what we call salts of these fatty acids. And so if we use sodium hydroxide, we get sodium salts. If we use potassium, we get potassium salts. I think someone once explained it to me that sodium salts or sodium soaps are hard soaps and potassium soaps are soft soaps. I don't know if that's necessarily true or what that means, because I don't know if soft and hard are like the same thing as like a liquid soap versus a bar soap, or if it just means the soap itself is either softer or harder. That one probably makes more sense. Sorry about that. Actually, I should probably look at that to see if it's important. No, not at all. Okay. Put that on silent. Alright, so that's how we make soaps, and they come from triglycerides. Now, let's talk about some complex lipids. So those are simple lipids, glycerol and some fatty acids. Now, lipids, remember, come from the term insoluble in water, basically water hating. And so there's some more. So we've got some phospholipids. Now, these contain glycerol, or an alcohol, or I'll write glycerol, but an alcohol, 2 fatty acids, and a phosphate ester. Now, I teach 2 classes that are going over the same material. Well, not the same type of classes. I teach Chem 170 also, which has a section on phosphate esters and functional groups. So I can't remember exactly which class it was that we focused on some phosphate esters. I'm going to guess that it was this class. So remember when we were talking about anhydrous or anhydrides, esters and amides, we did look at a couple phosphate esters because of their biological importance. And so that's where we're going to get to see them, is in these phospholipids. There's a couple different types of phospholipids. Phospholipids could contain glycerol. When they do, they're called glycerophospholipids. Okay. And when they don't contain glycerol, they contain a different molecule called sphingosine. And I want to say that in these sphingolipids, that it's sphingosine 1 fatty acid, and phosphate ester. So these are why they're called phospholipids, because they contain the phosphate esters, they contain fatty acids, and they either contain the alcohol sphingosine or the alcohol glycerol. So I probably should have wrote alcohol because it was a more general term. Okay, but I'm going to show you these structures. Don't worry. So kind of general breakdown here, not giving you anything real complex to look at. So we've got the simple triglycerides. My bad. Apologies. Simple triglycerides. We've got glycerol and 3 fatty acids. And then the complex ones, you can see I mentioned these already, the phospholipids. We'll come this way and look at the glycolipids in a second. Those do not contain a phosphate ester. In the phospholipids, we have the glycerophospholipids, which are just like glycerol, or just like our triglyceride, right? Except on that position where that third fatty acid went, we've got a phosphate group and we've got an amino alcohol, and I'll talk about some of those amino alcohols in a minute. In the sphingolipid, you can see here that it does indeed only have one fatty acid, and that's because, and they're not really doing a great job showing the structure here. If I was drawing it, I'd say that this is sphingosine, that this is my fatty acid, and here is my phosphate ester. And the reason that I would do that is because the sphingosine molecule itself does kind of have a big long fatty acid part that looks like the big fatty acid part. So it's similar to the glycerophospholipid in shape. It just doesn't need to have a second fatty acid attached to it. All right, and then the glycolipids you can see are sphingosine derivatives. They have a fatty acid on them, but instead of a phosphate ester, they're going to have a carbohydrate on them. And in most cases, it'll be glucose or galactose or some version of a modified glucose or galactose. I think N-acetylgalactosamine might be familiar to any of you that have studied peptidoglycan layers in bacteria. Biological membranes. So complex lipids from the membranes around cells and around small structures within cells. My bad. I said from. That doesn't make sense when you say from. Complex lipids form the membranes around cells and around small structures within cells. And in aqueous solutions, these complex lipids will spontaneously form, not from, into lipid bilayers. And these lipid bilayers have a back-to-back arrangement of lipid monolayers. And I have a picture that I'm actually going to show you. But I wanted to give you some of the components, okay, of these complex lipids that form the membranes. And they contain polar groups. They contain non-polar groups. These biological membranes is what we're talking about. The polar groups, we would say, are hydrophilic because they love water. And these are going to be what we call the head groups. The head groups of our lipids. So we're going to try to imagine, we've got glycerol, fatty acid, sphingosine. Phosphate ester, okay? So glycerol, fatty acid, phosphate, alcohol. So we're going to sort of picture this as a polar head group, okay, and a non-polar tail group. Okay, and so all of these little lipids, these little phospholipids, are going to start associating together, polar sides to polar sides, and non-polar sides to non-polar sides. And if you get enough of them together, they start to organize into these little, rafts. And if you let them, they'll close in on themselves like these little micelles. But if you have enough and they form this bilayer, you end up with all of the non-polar tail portions facing inward, okay? All of the polar head groups facing outward to the aqueous environment of the outside of the cell, right? Or the inside of the cell, which is also aqueous. But across this membrane now, things can be transported one way or the other, and we'll talk about how that happens. But we've created a barrier, a boundary. And the major force that's driving the formation of these lipid bilayers is those hydrophobic interactions, the interactions that are happening between those hydrophobic tails. And the arrangement of hydrocarbon tails in the interior can be really rigid if it's rich in the saturated fatty acids, or it could be more fluid if these have kinks in them. You know, if these are full of kinks, then they're not going to be able to pack together as tightly or as nicely as you can if they don't have kinks. And that's going to add or take out motion and fluidity within this bilayer. Let me show you a picture. So then you can imagine these things are moving up and down and sort of like waves, right? That can go through them, and all of these little, these proteins. So this protein, we don't see where it's going, but it's sticking through this bilayer, okay? And there's a little portion that sticks out on the other side. That little bit that I drew, that's what all of these things are. All of these things are membrane proteins that are spanning the gap. They span the gap because part of their protein structure is very nonpolar, and that part likes the inside of this membrane. And so when these things kind of float over here, they just kind of pop right through the middle and then hang out right there, where their polar bits are on the outsides, their nonpolar bits are right on the inside of this bilayer. And everything that can associate with a lipid bilayer is going to have that kind of arrangement. Non-polar insides and polar outsides so that it can fit in here. Well, anyway, these things can move around, forward, backwards, into the screen, out of the screen at us. And they do that because nothing is tethered to each other. They're all just next to each other because they're like little magnets. They're all interacting with each other. Okay? And the more you want these little rafts to be floating around, to be able to move through here, then you would want to have more fluidity. You'd want to have more kinks in your chains. If and if it's a hot environment and there's too much motion and everything's wiggling too much, you might want to have more structure, more rigidity. And so you would have to replace these with saturated fatty acids so that everything can kind of move slower and everything packs together better. And those are the dynamics of living systems. We're not going to talk about how they do that, or we will look at how cholesterol can add to the fluidity of these membranes. You can see cholesterol molecules here embedded in the structures, and those are again part of keeping this thing fluid or less fluid. All right, let's talk about glycerophospholipids. So glycerophospholipids, also called phosphoglycerides, are the second most abundant group of naturally occurring lipids. Okay, so these that we're looking at here, these are glycerophospholipids, or there's a great proportion of these are glycerophospholipids. The others are going to be just the phospholipids with the sphingosine in them. But they're found almost exclusively in plant and animal membranes. Typically consists between 40 and 50%. So the membranes are 40 to 50% of the glycerophospholipids, and about 50 to 60% proteins within or making up those membranes. Now, the most abundant glycerophospholipids are derived from phosphatidic acid. This is the molecule that we looked at earlier in a different chapter. The molecule in which glycerol is esterified to, and one of those phosphatidic acids end up forming that phosphate ester. The three most abundant fatty acids that are the phosphatidic acids are going to be palmitic, stearic, and oleic. These are the ones I showed you earlier. Most abundant. And you can see here a phosphatidic acid. The phosphate down at the bottom, oleic acid, palmitic acid. Now, the fatty acid on carbon 2 of glycerol is always unsaturated. And further esterification with low molecular weight alcohols will give a glycerophospholipid. So phosphatidic acid, when it comes in contact with the like I said, low molecular weight alcohols, they react together to form these glycerophospholipids. And glycerol is the low molecular weight alcohol of choice in our bodies. Now, the structure of glycerophospholipids is very similar to that of fats. The alcohol is glycerol. 2 of the 3 hydroxyl groups are esterified with fatty acids. The 3rd hydroxyl group is esterified with that phosphoric acid. And I have a picture. And you can see here, this is that phosphodiester bond. It's a diester bond because of the second one. At the moment, though, when it's just getting latched onto this glycerol, it's just esterified on one side, and it's just that phosphoric acid residue. And then it'll get esterified to another alcohol. And in a lot of cases, the alcohol is choline. And it's an amino alcohol because it has that amino group on it as well. Now, there are different kinds of molecules that we classify that are depending on which amino alcohol you get. So if you've got choline, then we call you a lecithin. And there are cephalins, which have different. I think they have serine, or what's the other amino alcohol? Triethanolamine. And so again, depending on which one you have, and also depending on whether sphingosine is attached to the phosphoric acid or if it's glycerol, there are different categories or classifications for those specific lipids. But they all form basically the same type of function. They're membrane functions, membrane molecules. Some of them are more prevalent in, let's say, nerve membranes or membranes in the brain, versus just everywhere else in the body. Okay. Here's a space-filling model of some complex lipids in a bilayer. As you can see, those hydrophobic tails point inward, the hydrophobic heads are on the outsides. Kind of gives you an idea of what this would look like. Now, cephalins, as I was mentioning, are the other type of glycerophospholipid in which ethanolamine or serine, what did I say? I said triethanolamine. I misspoke there. It's not triethanolamine. So ethanolamine, this other amino alcohol, and serine, which is also an amino acid with an alcohol group on it, form the cephalin groups. And so again, only difference here from a regular glycerophospholipid with what were we calling them? The ceramide glycerophospholipids is that the ceramide has a different alcohol here. So these are cephalins. The ceramide had it wasn't a ceramide. I'm even confusing that with another term. Sorry, ceramides I believe have sphingosine in them. So these are cephalins. The one that we looked at previous was a lecithin. A lecithin. I need to get that straight. So lecithins have the choline, and the cephalins have either ethanolamine or serine. And then I was talking about ceramides. I think we're going to get to those with sphingosine. So let's switch over to the sphingolipids. Now these contain a long chain amino alcohol, sphingosine, and so that's why they're named as such. The sphingolipid myelin, if you've heard of it, the myelin sheath, it's a coating found around nerve axons, and so sphingolipids make up a lot of those types of nerve coatings, the myelin sheath. And you can see that here. So here's sphingosine by itself. Here we get a ceramide. So a ceramide is when we have sphingosine, where the nitrogen, so that's something that's different about glycerol also, right? We had glycerol, it had its OHs. One of them gets attached to a fatty acid. Second one gets attached to a fatty acid. And then we get our phosphate and our alcohol. All right. In sphingosine, remember the top part sort of already looks like the glycerol with a molecule of fatty acid on it. But then instead of getting that fatty acid attaching to the alcohol group on sphingosine, it's worth noting here that it's on the amino group. So that's where they're different as well. So in sphingosine, this is a nitrogen, or an amide. Okay, that's why they're called ceramides, a ceramide. They're called ceramides because they're amides, but you have to have sphingosine to get the amide group. So, and so then you see here the addition of the phosphodiester bond and the what is this guy? Ethanolamine gives you sphingomyelin, and like we were saying, sphingomyelin is found in the myelin sheath, helps to insulate nerve transmissions. Glycolipids, we didn't spend much time talking about. Glycolipids are complex lipids that contain carbohydrates. Carbohydrates and ceramides. So carbohydrate, you guys would know monosaccharides are what we're talking about. Glucose or galactose. Okay. The cerebroside. There's a compounds called cerebrosides, and these are ceramides that are found in the brain, hence the cerebro. Ceramides that contain mono or oligosaccharides. So they can have one sugar on them or multiple sugars, oligo meaning many. The following molecule, or the one I've got on this screen, is a glucocerebroside. So it's a cerebroside that contains a glucose. And you can see here is the sphingosine, right? Sphingosine. It's got an R group right here, so it's got a fatty acid on the amine, so it's a ceramide. And then in place of a phosphate, it's got a sugar. And in this case, it's glucopyranose or glucose. And so this is a glucocerebroside. But again, that's just some of the naming. Carbohydrate-containing ceramides, or a glycolipid. So let's switch over to steroids. So steroids are a group of plant and animal lipid that contains a tetracyclic ring. And it's this setup. So six-membered, 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 though they're not in a row, okay? And then a five-membered ring. Now, specifically, this is what cholesterol looks like. Cholesterol is the most abundant steroid in our bodies, and it's the most important. It's a component of the plasma membrane, as we talked about earlier, helping with keeping it fluid. It's also a precursor of the steroid hormones and bile acids. So here's a ball and stick model, and here again you can see the skeletal structure, right, of steroid. So you should be able to recognize that in all of our steroids. So cholesterol is the starting material for the synthesis of steroid hormones, and it's converted first to progesterone. And so you can see, well, I don't have the structure of this guy still up. You can see it had a couple methyls, a hydroxyl, and then this little bit over here. And most of that's still true. The methyls are still there. This bit got changed, and now hydroxyl got oxidized. So it gets turned into progesterone. From progesterone, it can then be converted into both the sex hormones and adrenocorticoid hormones. And so you can see here progesterone into testosterone and estradiol, and then it can also go into cortisol, cortisone, and aldosterone. And we'll talk about a little bit of what these hormones do in a few minutes. But you can see definitely through a series of reactions, right? That cholesterol is the main precursor to this. Now, lipoproteins carry cholesterol. Okay. Most lipoproteins contain a core of hydrophobic lipid molecules surrounded by a shell of hydrophilic molecules such as proteins and phospholipids. And there's 4 kinds of these lipoproteins. Now I'm showing you a general, this is an LDL right here, but you can still see some of those bits. We've got phospholipids on the outside, we've got proteins on the outside, and then for the most part, the cholesterol, which is nonpolar, is going to be found on the inside here. They don't all look like this. LDL happens to carry more cholesterol than others, but let's talk about what they do. So we've got HDL, which is a high-density lipoprotein, or good cholesterol. This has about 33% protein and about 30% cholesterol. We've got LDL. This one has about 25% protein and 50% cholesterol. So it's just carrying more cholesterol in it. Okay. We have VLDL, very low density lipoproteins. These carry triglycerides or fats that are synthesized by the liver. So they carry those to different parts of the body. And then we have chylomicrons. And these carry dietary lipids. That are synthesized in the intestines. So chylomicrons carry the fats that you eat around your body. Very low density lipoproteins carry the triglycerides that your liver makes, either to storage or back to some other place for energy if you need energy. You've got low density lipoproteins that are responsible for sort of transporting the majority of cholesterol, and then you've got high density lipoproteins, which are sort of thought of as the scavengers. They go and scoop up cholesterol from the blood and from the cells, and they bring it back for packaging. I believe that happens also in the liver. And so they work, all of these different lipoproteins are working in concert to sort of make sure that the lipids, which wouldn't dissolve normally in your aqueous blood, right, get transported everywhere they need to go. Now, if you have a lot of cholesterol, then you're going to have a lot of it floating around in your blood. Now, having a high HDL count is good because that means you're going to have, scavengers running around cleaning up that cholesterol. If you happen to have more low density lipoproteins than high density, then not only do you have a lot of cholesterol floating around, but you're also still transporting a lot of it around. And most of it is that you make. You don't get most of the cholesterol from your diet. You make most of it. But it can build up in your arteries, and that's the bad thing. But anyways, that's what these lipoproteins do, what they are. And I guess I was going to tell you all about this stuff anyway. So what I was saying is transport of cholesterol starts in the liver, or I guess I should say the liver is the source of your cholesterol. And it starts out as a very large VLDL, very low density lipoprotein particle. And this is carried out, so we're talking about cholesterol, from the liver in this VLDL into the serum, your blood. And as fat gets removed, its density increases. And that very low density lipoprotein actually starts to become a low density lipoprotein. And a low density lipoprotein stays in the plasma for about 2.5 days, floating around, doing its job. Now, LDL carries cholesterol to the cells, okay, where specific LDL receptors on cells will actually bind to it, and they'll transport cholesterol to each other through these through these specific, after binding, the LDL is taken into the cells. Actually, the whole molecule gets brought inside the cell. So if this is an LDL molecule, it gets brought in through a process called endocytosis, or we're not going to go over that. It gets brought in, and then it liberates the free cholesterol and the cholesterol esters. Sometimes cholesterol is transported as a cholesterol ester. It makes it more soluble. And so they get transported. And so then high-density lipoprotein transports cholesterol from these peripheral tissues. So now HDL comes and picks up that cholesterol and takes it to the liver. And it also transports cholesterol to the LDLs. So while in the serum, free cholesterol that's in the HDL is converted into cholesterol esters. Okay, in the liver, HDL binds to the liver cell surface and transfers its cholesterol esters to the cell. So it's really transporting it as the esters, and it gives it back to the liver. Those esters are then used as the synthesis of steroid hormones and bile acids and everything else. And then after the HDL has delivered its cholesterol esters to the liver, it re-enters circulation. Okay, so back here. So that's sort of the transport. Now, most of the cholesterol is carried by LDL. Normal plasma levels are around 175 milligrams per 100 milliliters. And if there are sufficient LDL receptors on the cells, then the LDL is removed from circulation and its concentration drops from in the blood. But the number of LDL receptors is controlled by a feedback mechanism. So when the concentration of cholesterol inside the cell is high, then the synthesis of those LDL receptors is suppressed. The cells aren't showing as many receptors because they don't need any cholesterol. They've got high levels already. So there's this disease called familial hypercholesterolemia, and there's not enough LDL receptors, and so the plasma levels of cholesterol are as high as like 600 milligrams per 100 mils. And those high levels of cholesterol can cause premature atherosclerosis and heart attacks. And in general, high LDL means high cholesterol content in the plasma because LDL can't get into the cells. So high LDL together with low HDL is a symptom of faulty cholesterol transport and a warning of possible atherosclerosis. The serum cholesterol level controls cholesterol synthesis in the liver. And so when serum cholesterol level is high, the synthesis in the liver is low and vice versa. The commonly used statin drugs inhibit the synthesis of cholesterol by blocking HMG-CoA reductase. It's an enzyme that's part of that cycle. All right. Cycle of cholesterol synthesis. Kind of lost what I was thinking. So here are the male sex hormones synthesized in the testes from cholesterol, responsible for the development of male secondary sex characteristics. You can see there testosterone and aldosterone. Among the synthetic anabolic steroids are these guys: methandione, methanolone, 4-androstenedione, 317-dione. These are synthetic anabolic steroids, but you can see by the structure why they are called steroids. Now, the female sex hormones, these are synthesized in the ovaries from progesterone, responsible for the development of female secondary sex characteristics, and they control the menstrual cycle. You can see here the similarities between progesterone and estradiol. Now, progesterone-like analogs, so compounds that look like progesterone, these can be used as oral contraceptives. So here in norethindrone, nor just means the absence of that methyl group that is there in our cholesterol and in this other molecule. Because they share those similarities in the structure, we can, and that's kind of how medicine works, is you just use molecules that look similar to other molecules and hope that the body wants to do something with it, and it's guess or check. Sometimes they're harmful, sometimes they're helpful, sometimes they don't do anything. But the more we know about the structure and function of molecules in our body, the better we will be able to build molecules to do certain jobs, certain tasks. So the glucocorticoid hormones. These are synthesized in the adrenal cortex. So on the outside of the well, I want to say of your kidneys, but I think there's something on top of your kidneys, your adrenal glands, right? I think the cortex is like the outside of it. No, it doesn't matter. But these are synthesized in the adrenal cortex. They regulate metabolism of carbohydrates. So whether or not you're releasing insulin or whether or not you're, what's the other one, glucagon, or this, again, will have effects on intermediates in the glycolysis and in the citric acid cycle. Those are the systems that oxidize carbohydrates for energy. So they'll regulate that. Aldosterone is one of those. They also decrease inflammation. They are involved in reactions to stress. Yeah, glucocorticoid hormones. I mean, there's more things that they're responsible for than just that. Bile salts. These are oxidation products of cholesterol. These are synthesized in the liver. They're stored in the gallbladder. So if you don't have a gallbladder, you don't have a storage of these, but you still make them. They're secreted into the intestine when you are eating, and they help to break down fats because they emulsify fats, meaning they surround the fats and provide a part for the fat to stick onto, like a non-polar bit, but also have enough polar bits on them that water likes it. So if they can surround your dietary fat, they can aid in the absorption and digestion. And so both of these bile salts are, again, oxidation products of cholesterol, but yeah, you don't need to know the structures or anything. Now, let's talk about prostaglandins. These are a family of compounds that come from the 20-carbon skeleton of prostanoic acid. So here is prostanoic acid. And actually, there's another one as well. I think it's arachidonic acid. I think I have a picture. Yeah, there we go. Arachidonic acid. So this is another one. But again, prostaglandins, these aren't stored in the tissues as this guy. They are stored as arachidonic acid. They're synthesized from polyunsaturated fatty acids in response to specific physiological triggers, and so one of the ones that gets formed is this arachidonic acid. This is a prostaglandin. Again, it's a polyunsaturated fatty acid, and so just with the bonds, just the way that they've drawn them here, you can see that there's some multiple cis double bonds in there. But anyways, these prostaglandins are involved in pain and inflammation. And so some of those non-steroidal anti-inflammatory drugs, NSAIDs, they interrupt the synthesis of the prostaglandins that are ultimately released when you injure yourself by blocking, in this case, by inhibiting this particular enzyme, but by blocking some of the steps in the synthesis of that from the arachidonic acid. So again, the prostaglandins are molecules that get released that trigger things like inflammation and pain, and we can block them by inhibiting their conversion. Let's see. Thromboxanes are also compounds. Let's see. Thromboxanes are derived from arachidonic acid as well. Thromboxane A induces platelet aggregation and vasoconstriction. And aspirin and other NSAIDs also inhibit the synthesis of thromboxanes by inhibiting something called the COX enzyme, COX, cyclooxygenase. And this occurs in two forms. There's cyclooxygenase 1, it catalyzes the normal physiological production of prostaglandins. And then there's COX-2, which is responsible for producing prostaglandins in inflammation. So like when a tissue is injured or damaged, special inflammatory cells will invade the injured tissue and interact with the resident cells and such. And so then these COX-2 interactions form these other prostaglandins. But thromboxanes, like prostaglandins, or like the arachidonic acid compounds, are all involved in those pain responses. And lastly, we have leukotrienes. Leukotrienes are also synthesized from arachidonic acid. They occur mainly in leukocytes. These are blood cells. They produce muscle contractions, especially in the lungs, and thereby cause asthma-like attacks. In this regard, they're about 100 times more potent than histamine, these leukotrienes. And several recently developed anti-asthma drugs inhibit the synthesis of leukotrienes, and not just attacking histamine as an anti-allergy medicine, but attacking leukotrienes as well. All right, well, that's about it for this chapter. So I hope you guys learned something about lipids. Remember, there's a lot of different kinds of lipids here. We're just focusing on some of the different classes and some of the differences between them. But if you guys have questions, don't forget, email me, post a discussion, and if not, I'll see you guys in the next video. All right, take care, everybody.